Thanks, Jen, and thank you very much, Dr. Johnson. Living, in the pockets of proteins and the details of early drug development, I cannot remember having a chance to zoom out and learn about disease trajectories over populations and species. That was just delicious. Maybe not the bat surveillance slide, which will haunt me in my dreams tonight, but we so appreciate your leadership in this critical domain. Thank you all for joining this energizing, I hope also informative, first NSOF symposium on the most urgent and important topic of coronavirus antivirals. Personally, I found the ensemble of deeply considered biochemical and biological methods, machines, and medicines both educational and also just hugely energizing. It's fair to say that in this present moment of biomedical research where we have an atomic resolution of disease biology, we can imagine, invent, and translate medicines with just unprecedented speed and sophistication. Today, we learned the performance characteristics and predictability of preclinical model systems of COVID-19, still an area of vital need and facile access. We functionally and structurally resolved divergent blocking biological therapeutics presently in clinical study amidst viral variation and natural selection and any number of challenges. We evolved beyond reconsidered legacy chemical matter towards intentional, direct-acting, low molecular weight pan-coronavirus protease inhibitors that will surely benefit from computational guidance, anticipating further emergent and treatment-resistant variants. We celebrated the innovation of remdesivir, which in my opinion stands as just a historic campaign in wartime drug development. We've also been thoroughly introduced to nuanced mechanistic distinctions of next-generation RDRP inhibitors, and we looked around the corner at next-validated targets, explored in a real chemical biology mindset, again, powered by high-resolution structural biology, RNA methyltransferases, the 2' methyltransferase, the NSP13 helicase. Importantly, these ideas were shared by a highly diverse audience. Therapeutic science requires this diversity in every dimension, and it's so wonderful to have at one table academic, biotech, and pharma scientists, chemists, biologists, physicians, legendary, experienced professional scientists, and rising rock stars like Ashley, who gave just a stunning presentation on 189010, the RDRP NIRAN domain, which was not previously on my radar, but is now rocking in my PyMol browser. Okay, 4 thank yous, 4 challenges, and 1 painless favor. Thank you to our speakers for exceeding our request for hard-hitting science without any thinly considered panels. You are awesome. Thank you again to our organizers and session chairs who created what I think is a really important gathering that brings together drug hunters from around the globe. Thank you to all the research scientists at NIBR and beyond who bravely and selflessly took to laboratories, just like clinicians at the bedside during an uncertain and surely frightening time to respond to COVID-19, but also cancer, neurodegeneration, and thank you to all who joined today, as well as those who will join in the future as the speakers have graciously permitted the preservation of this bookmark in coronavirus therapeutics as an open and online resource. The impossible challenge of these 18 months can't be overstated, and the suffering and loss just can't be forgotten. But in this moment, I can't help but be hopeful. So thanks for your ideas and your conviction. Now, it's not my nature only to compliment and celebrate. We have a lot of work to do. So 4 pieces of critical feedback for consideration from this community. First, we have to be better prepared for the 8th zoonotic transmission with community spread, as Dr. Johnson rightly warns. Let's please commit to carrying this research as far as it can lead us, even as the risk of SARS-CoV-2 dissipates and other priorities seem more pressing. In truth, my greatest hope for our own work at NIBR on MPRO is that the work continues to move quickly and authoritatively, but is ultimately unnecessary. In this instance, we will be better prepared and so much better connected for the next pandemic. Second, we have to skate to where the puck is going. These coronavirus medicines will ultimately be used to benefit patients only in combination, and we have to organize to study antivirals in appropriate preclinical combinations to pick winners bravely, even if it's not our own medicine. Realistically, then, to prioritize a very small few for study in these hopefully waning moments of SARS-CoV-2 so that we're better prepared next time. Third, we can work even better together. Access to models, directional preclinical models are challenging to access at scale, and giving actionable data at dose remains a challenge. Use of protein structure. Coordinates have been a massively accessible resource during the pandemic, truly explanatory, but not as yet guiding therapeutic discovery, but for a few exceptions like the main protease. Collaboration. Through this time, we've importantly created bonds of friendship, scientific and personal adjacencies. Surely this has been true among many, but not all, large pharma heads of R&D. But some defenses are hard to surrender, and barriers to collaboration in preclinical research, and especially in early clinical investigation, still exist. And we see that everywhere, not only in coronavirus. Just think what we could do for other serious diseases. And last, we just must do this together sometime, face to face. Information transfer is highly effective and massively inclusive over the airways, but energy transfer is inefficient. So we'll plan for a hybrid experience next year. With all that said, what a time to be a scientist. 2,000 coronavirus structures in the PDB, 100,000 coronavirus contributions to PubMed, 2 billion doses of vaccines administered in 18 short months. A first low molecular weight antiviral fully approved, blocking antibodies, accessible by emergency use authorization, and so many more medicines on the horizon. We look forward to seeing you next year at the second annual Neuroscience of Therapeutics Symposium. Now the favor. This NSOFT is a new concept for us, a massively open online symposium, if that's a term, in the traditionally highly private and proprietary space of drug discovery. We are going for more of a Gordon conference than a Novartis corporate event. And we want to know, what was your experience? What's your advice for next time? We genuinely welcome your feedback, and if you can, please stay with the event for just a moment after we close and contribute your reflections and guidances via quick and, I promise, painless survey. Best wishes. Take care of yourselves and each other as you work to save humanity. Goodbye.